In this video, we'll talk about ion trapping of drug. Let's understand this with the help of an example. Say a patient has consumed a tablet of aspirin. As soon as it reaches the stomach, it disintegrates in the stomach into ionized and unionized form. Now, how much of the person aspirin will remain in ionized form or in unionized form, that can be ascertained with the help of pK of the drug and pH of the medium. That means if you have these two parameters, then you can easily estimate how much of the aspirin will be in unionized form in the stomach and how much will be in the ionized form. With this formula, you can easily calculate the percent ionization. So for example, pK pK of the aspirin is 3.5 and pH of the medium, so stomach pH is 2, so it will come around 3%. That means only 3% of the drug will be ionized, rest 90 approx 97% will be unionized. That means 97% of the aspirin in the stomach will be available in lipid soluble form, while rest will be available as water soluble form. However, both tries to get across the cell membrane, but ionized being water soluble cannot cross the cell membrane, whereas the unionized particles of the aspirin can easily cross the cell membrane and enters the cell lining the stomach. Now, here is the catch. Here, within the stomach lumen, the pH was acidic and our drug was also acidic. That means when the medium is same, most of the drug remain in unionized or lipid soluble form. But as soon as aspirin enters the cell lining stomach, the pH of the stomach cell is almost neutral, 7, which is relatively alkaline with respect to pK of the drug, that is 3.5. In this case, since the medium is different to the pK, most of the drug will remain in ionized form. Now, the same thing with the help of this example we can understand. Here, the pH of the pK of the drug is same 3.5 and pH of the stomach cell is 7. As a result of which 99.99% of the aspirin will be ionized. That means 99.99% of the aspirin is water soluble in the stomach. Remember, this was earlier the case. In the lumen, most of the aspirin was in unionized form. However, this being lipid soluble, as soon as it crosses the stomach cell, within the stomach cell, when it gets to neutral environment, it converts itself into ionized form. Earlier, the same thing in the stomach lumen was unionized or lipid soluble, but now it is water soluble. That means these ionized particles of aspirin cannot further cross the cell membrane, neither it can come to the stomach lumen. However, the rest unionized or almost less than 1% of the unionized particles of the aspirin is lipid soluble and it crosses the cell membrane and enters the bloodstream, and thereby it shows its action. Second, the leftover ionized aspirin particles will go into the small intestine from where it will be absorbed. Okay, earlier the ions were able to cross the membrane, but now it is unable to cross back to its original environment or the further next environment. The ions are trapped inside the stomach cell. This is called ion trapping. Now, if the aspirin remains as ionized form within the stomach cell, there is a high chance of toxicity. This is the reason if we continuously take aspirin, the chances of stomach ulcer is too high because it inhibits the COX enzyme of the stomach cell. Okay, this COX enzyme is responsible for the production of prostaglandins. This prostaglandin protects the lining of the stomach and it do not allow the stomach acid to come in direct contact with the stomach cell. However, if there is too much of aspirin in the cell, which prevents the formation of prostaglandin and stomach thereby less protection. Hence, the patient may suffer from stomach ulcer. Apart from this, the body does have its own mechanism to efflux this ionized aspirin from the stomach cell. The initial absorption of aspirin from this cell membrane was by passive diffusion. It is energy free. However, now when the ionized particles need to be effluxed from the cell, it is totally energy dependent and active transport and via ion channels these are released from the cell. Okay, this is all about ion trapping. Now, ion trapping do have a crucial role in toxicity case. Say for example, if a patient has consumed a toxic and which is basic in nature, say for example, this is amphetamine. There is amphetamine toxicity. In that case, of course, if the period of exposure is less than 3 to 4 hours, then we can go for induction of vomition. But if it is more than 4 hours, and what if the compound is already absorbed in the circulation? In that case, we can go for rapid elimination of the amphetamine from the urine. So for that purpose, since amphetamine is basic drug, we can give acidic compounds such as ammonium chloride to increase excretion of the amphetamine. When the medium is safe, absorption is good. When the medium is different, excretion is good. Here, we are giving ammonium chloride to make the urine pH acidic and compound is alkaline. That means there is difference in the medium and the pK. So, elimination will be faster. Same is the case in case of aspirin toxicity or any acidic drug toxicity. We can go for bicarbonate to make the urine alkaline. Since the drug is acidic, urine is alkaline, excretion will be faster. So this is also ion trapping. So this is all about ion trapping.